I guess people talk a lot about post-pandemic, and I don't think anyone's told the virus that it's post-pandemic. So I feel like we're all really tired, and we all want our lives back and normalized again, on the one hand. And on the other hand, I don't think this virus is done evolving. And the way it is selected is for being either more infectious or being resistant to immune responses. Those are the two things that put pressure on the virus to change.
Bette Korber is a laboratory fellow in theoretical biology and biophysics at Los Alamos National Laboratory. She's lauded for her pioneering achievements in virology research and computational vaccine design, which provide hope against diseases such as HIV/AIDS. Korber delayed retirement in 2020 to study SARS-CoV-2 and soon demonstrated that the virus was rapidly evolving to become more infectious, a discovery cited literally thousands of times. Her numerous honors include the Ernest Orlando Lawrence Award, the Department of Energy's highest scientific honor, and both the Los Alamos National Laboratory's Richard P. Feynman Innovation Prize and Los Alamos Medal, the lab's highest honor. Welcome, Dr. Korber. Congratulations, and thanks so much for joining us here at the beautiful Caltech Alumni House. Your work in virology and vaccine design has been groundbreaking, and it's going to be exciting to dive into talking about that this hour. But first, one of the wonderful things about our Caltech Distinguished Alumni is they are often multifaceted, and you yourself are a singer, songwriter, musician. Your award-winning song, Veneration, is a beautiful song about the beauty of nature, and you dedicated it to your parents, George and Gladys Korber. And can you tell us a little bit about your parents, growing up with them, and the adventures that you all had?
Sure, yeah. So I grew up in Long Beach, and I'm Angelino, for the first 30 years of my life through my time at Caltech. But my parents were ranchers from Montana. Originally, that's where they grew up, and they moved here to get away from the harsh winters. And my family was not really a college-bound sort of family typically, and my dad had wanted to go to college more than anything when he was growing up on the ranch. And so he was in World War II, and he had the GI Bill, and he got to go on through Stanford and got a PhD in race relations. He wanted to understand why we don't get along. And they were wonderful. And so we did spend summers, long vacations. He was a Cal State professor, and we would spend long summer vacations going to Montana, and we would always go through the national parks, and we would spend time in the parks. And so I think that's where my love of nature came from. Was my mother and father both really, really love nature, and they were disappointed when I wanted to go into science.
Really?
Yes. They wanted, my father wanted me to go into journalism. I really like to write, and he felt that was the best way to impact the world. And I kind of convinced him as time went on that, I think both journalists and scientists love and seek truth, and it's hard. It's often gray areas, but both fields try to find those things and express it for society. And so he reconciled eventually with the career choice that I made.
Yes, and he would be very proud of the impact that you've made. So your interest in science, so it began, tell us about that, and then how that led you to Caltech eventually.
I really loved chemistry when I was an undergraduate. It was beautiful to me, and I had some aptitude for it, and I had no idea I was going to be a scientist. I did want to be a writer initially. So I just marveled at what I was learning, and I loved my undergraduate science classes. So I switched majors from English to science, and that, I guess that's where it began. So I didn't always know I wanted to be a scientist, or I didn't even have any idea that I did. It was just being fascinated as a student.
So you, at Caltech, and you've had a number of mentors at Caltech that were very important to you.
Well, I did, yeah. And it would be nice to talk about a couple of them. Actually, I came in as a chemist, but there weren't many women in my department at that time. And in fact, several of them didn't make it through. They dropped out. It wasn't easy, actually. And
Can you break that down a little bit when you say it wasn't easy? Was it not easy being a woman?
yes, I think specifically. And so things happen. Like I had come in intending to do sort of more theoretical and mathematical chemistry, which is what I liked in my undergraduate work I'd been in. But I was given an advisor when I came in who thought that I had a terrible deficit in biology, which I did, virtually none. And so he said, he crossed out my schedule with a big black Sharpie, and he said, "You're taking biology classes." And I wasn't happy. We fought a lot about it, but I wound up, and I was intimidated, and I followed his lead. But as it turned out, I had a wonderful class with a woman named Ellen Rothenberg, who's still a professor here in immunology, and that was also really beautiful to me. And she taught with just such enthusiasm. She clearly loved the science, and it was contagious. So,
Contagious.
yes. So I learned to really appreciate the immune system from her and the beauty of it. And when you think about immunology, you sort of also think about evolution, and I think that also set a path to think about evolution for me. But so she was really important to me and really helped me begin a career. And then I wound up going through 2 labs, but in my second lab I was with Professor Lee Hood,
Yes.
who no longer at Caltech, but was a great scientist. He had a huge group that was really lively. So there were close to 100 of us, I think, at the time I was in his group. And the way that was handled was kind of a structure and a hierarchy, and I had a wonderful mentor there, Ivona Stranowski, who was sort of, there was scientists who were after their postdocs, but they were under Lee's umbrella at grants, and they would often get graduate students assigned to them. So Ivona looked after me and kept my science rigorous and helped me maintain a good path. So she was also really pivotal. And 2 other people, I think, gave me a sense of dignity when it was a struggle. And it wasn't easy being a woman in this context. And they were both men. They were great feminists. And one of them was a gentleman named Ray Owen. So he retired the year I got here. But he just loved students. And he was really so thoughtful. And he always would be putting the key articles that I needed to read in my mailbox. And then this continued for years. When I did a postdoc at Harvard, letters would come from Ray with articles in them that I needed to be reading. And this continued when I got to Los Alamos, and he was just magnificent. And so we would have lunches at the Ath every week, and he really wanted to see women succeed. It was important to him. So he especially nurtured us and gave us a lot of respect. The other person that I felt was really meaningful to me was actually Dick Feynman. I took a class from him called Theoretical Limits of Computation. And he was friends with my housemate, Brian Moore. So Brian was another graduate student and an exceptional physicist. And he and Dick had been in plays together, and so we would go out to lunch. And he would ask me about my science with such intensity and interest that I felt that it meant something. I felt dignified in knowing who he was, that he would do that. It really meant a lot to me. So he was another person who sort of gave me strength to get through by just respect and kind of a sweet respect, a kindness.
Yeah, and this is jumping around a little bit, but you've described yourself as you feel like a scientific grandmother.
Yeah.
And so, and you were talking about the role of mentorship, and so what do you mean when you say that phrase?
Well, I'm now in my 60s, and I've really been fortunate to have a lot of young people that I've been able to work with through the years that I've watched grow into just being fantastic scientists. So that's part of it. But also, I guess of a basic kind of concept, which is databases that we all share. And when I went to Los Alamos, I went there to work with a gentleman named Jerry Myers, and he had founded an HIV database at Los Alamos. And a lot of people don't really know this about Los Alamos, but it was one of the first theoretical biology groups in the world. There is getting to be enough DNA sequence that you could study it mathematically and you could look for patterns. So a couple of physicists at Los Alamos founded a theory group, and they're also studying the immune system dynamically and with equations, right? So that was a place where you could do this kind of thing instead of just doing experiment, which is traditional biology.
Right.
And when I went there, there was no internet, right? So it was, we would make hardbound copies of books.
About what year was that?
So that would have been 1981.
Right.
And so, I'm sorry, no, I'm saying it would be 1991. Yeah, 1991. So we would ship these books off to everybody's desk who was in HIV research. But as the internet was blooming, I wanted to get everything accessible to people online. So that became kind of a mission of mine. And I was growing up as a postdoc in this environment where GenBank had been founded, which is the repository for all sequence data of all organisms. It moved eventually to the NIH, but it was founded at Los Alamos. And my mentor was working on HIV, and I wanted to make ways that people could access this data without having to thumb through their books. And so I feel like I'm also kind of a scientific grandmother to the idea of sequence databases online. We have the first pathogen database, and I founded the first immunology database. So I'm kind of a grandmother of this kind of concept, I think. Yeah, and that I love having been there and been able to be part of that expansion of knowledge and that sharing of knowledge.
Yeah, and your work is really, I think you've talked about seeing your work change over the years, of the nature of what you're doing. Can you talk about that a little bit, like when it started and when it went?
Yeah, so when I did my thesis here, I did a little bit of sequencing of the interferon regulatory elements. And it was just a small bit of sequence, and it was years of labor to make it and create it and interpret it. And by the time I got to Los Alamos, there was a lot of HIV sequences available, and that's why we have this database. And then there started to be this sort of global collection and efforts, and the sequencing technology got a lot better, part of which was done in Lee's lab here at Caltech. He really worked on innovative strategies for sequencing to speed up the whole process. And so things just kept growing, and this HIV database that we created is still thriving and is still a major resource for researchers all over the years. But when SARS-CoV-2 hit, and we wanted to be able to look at that data, everything changed again. So this is just 2 years ago, and there was a group called GISAID, and this is an organization that does data sharing. So they started sharing sequence data from this virus, and every sequence from the virus would come from someone getting a nose swab, and then they would instantly be able to do the sequence. So the technology had rapidly changed. They would link that to a location and a date of sampling, and maybe some other information, some called metadata, like their health status or something. That wasn't essential. The date and the place was really the key thing. And they would put it in this repository called GISAID, and now we have 11 million SARS-CoV-2 sequences available. And they're available from a week ago, 2 weeks ago. You can get fresh data from all over the globe. So it's been something that people all over the world have contributed to try to track the evolution of this virus and how immune pressure is causing it to evolve and escape. We can understand that because everyone contributes freely to this database to share. And when I work in the HIV field, our sequences are often 2 years old before we see them. There's a wait until you publish,
Right.
wait until it's reviewed.
Right.
This is not what happened with SARS-CoV-2. Everyone gave instantly. And to me, this was a revolution in the way we were doing science. And it was controversial to begin with because we were used to the more standard databases. You have to do a data sharing agreement where you respect the sort of propriety of the person who contributed the sequences. But it's really almost been miraculous to me that we've been able to study this virus in this way. And it's kind of an unsung story because we just assumed that we're going to know, is the vaccine going to work against this variant? And is it going to work against that variant? And how is it going to impact the way we do healthcare and public health? But we wouldn't have known it without the system changing and without it being so enabling. So went from my graduate work where, a few thousand bases were 3 years of work and very difficult to get out, to now 11 million sequences of this virus. And now we have monkeypox breaking out this week, and instantly everything is getting sequenced. It's a huge genome, and it's like this, and everyone's sharing the data, which is kind of a new paradigm. We're not waiting for the publication, we're sharing. And it's really beautiful and amazing to me.
Well, it's interesting. I mean, did anything surprise you when you saw your sequence with the COVID? With anything surprised you in the data that came in?
Oh, absolutely. So I think HIV evolves incredibly quickly,
Right.
and so does hepatitis C and other viruses that I've studied. And when this one came out, it had a proofreading mechanism, so it doesn't have quite the same error rate. And it led people to assume that it wouldn't be evolving. And this was a virus and a new host. And I wanted to look for evolution. I wanted to see if I could see patterns. And so if you go back to the newspapers or the press in early 2020,
Right.
there's a lot of declarations about this virus is not evolving. There's many. So it's going to be fine. The vaccine will always work. This is got a low error rate because so we don't have to worry about this. And traditional methods that we have for looking at evolution in something like HIV that evolves really quickly were not the right way to look at the question. So as soon as I started looking at the data, I started looking at it more like kind of classic proof of Darwinian evolution. A story, if you have this mutation, or if you don't have this mutation, how does the virus change in its growth rate? And because of GISAID, because of this remarkable database, I was able to do this in every state, every country, to the city level. And so I could see that there was a mutational pattern in the virus that didn't show up initially. The Wuhan form of the virus spread. And then this pattern, if it was introduced into the community within 2 weeks, it was becoming the dominant form in that community. But it was only a couple base changes, so it wasn't like it was a repeated pattern of evolution. It was a form that was more fit. And so it was a competition, and the more fit form would take over. And every place it seeded, this would happen within a month. But meanwhile, everyone was declaring the virus wasn't evolving. So this was surprising, and it was not readily accepted. It was very controversial. So I submitted a paper in bioarchives in March saying this virus is evolving, and it's getting more fit, and it's getting more contagious. And it's really rapidly changing all over the globe to this more contagious form. And it was one of the most downloaded papers that bioarchive had at the time. It was hundreds of thousands of downloads and protests. So the scientific community said, no, she's wrong. But now they know that it's correct. And so I eventually published this in Cell, and it's probably my most cited paper. But it's certainly, everyone knows it's changing now. You guys all hear about the variants.
Right.
And the variants can change globally, shift from one form to another in a period of just 1 to 2 months. I mean, the whole global framework of thinking about the virus has to shift, Delta and Omicron. Well, this was the first one of those, and it really made a transition in about 2 months, and it showed how quickly this virus could change its phenotypic nature in just a 6-week period. So I think that was a really important thing. And as I was doing this, because I had worked on databases and HIV databases and immune-driven evolution, I knew we were going to need to look at variants. And so I think once people realized it was evolving, a lot of people got on board, and there's many great groups doing this in the world. But I've gotten to be part of something called the SAVE Project, which is the NIH group, and I'm in their early detection units. So we kind of probe gizzade and we look for new variants. And as soon as something arises, whether we find it via scientific posts from our colleagues who have it in their laboratory or whether we find it in gizzade, we alert the immunologists and let them know this is the new variants that are coming up. We don't know for sure often when they're just beginning which one's going to take off, but we try to get those on people's lab bench so they can start studying them immediately. And then when one does take off, they've got the framework to be able to think quickly about what the immunological consequences are. The vaccine's going to continue to work? Are there going to be any issues with being still sensitive to drugs, that kind of stuff?
I mean,
so that's what we do now.
So to go back to the beginning of COVID breaking out, and and you're saying in the news certain things are being said.
Right.
It's not changing, and the science is different. And of course, if your background of a journalist and a scientist, is there sometimes a tension between the fast-breaking news and the real science, as you see it?
I think everyone is doing their best, but the problem is the science changes quickly. And so it's been kind of an interesting evolution. We have now something called archives, which are ways to pre-publish. And so that's a way to get news out quickly. And those accompany a traditional scientific paper and don't block it. So while the traditional paper's under review, you can have your paper read. But there's been also a lot of tweeting with SARS, and that's new to me. I'm not used to this. And it's a mixed blessing, because oftentimes someone will discover something new that's really useful and get it out almost instantly. But other times people will just speculate on their tweets without, referencing citations to other people's work, without a framework, and it is just speculation, and it could be right or wrong, and that gets picked up by the press. And then it confuses people because the story changes. So it can be really, it can go either direction. And I do think the reporters are trying to get to the public what they need to know.
Right.
And they want the hottest, newest stories, so they follow the tweets. And the tweets, there's no sort of cost to a mistake in a tweet. You can speculate all you want. And many of my very excellent peers do that. and I know it's speculation, but it's not always delivered that way. So, so when things are still unsettled and we're still learning, sometimes there's, I think, been stories out there that are still controversial or not solved yet. Well, my own story was one of those stories because it was quite controversial.
Yes.
Many people disagreed with it. They came round, but initially they did. So I think it's difficult for the public then, because they don't, why are they changing their mind? Well, they're changing mind because data is coming in swiftly, and the public is now learning things very early on the curve of, as we're just beginning to understand things, they're getting informed. And it's confusing to people, and I don't think we've settled this yet. I think we're in the process of learning how to do this better. Maybe we won't ever get so good at this. I don't know. The information can move so swiftly now, and we're not accustomed to it, and the press isn't accustomed to it, and I don't know how it's going to shake down in future situations.
Well, we're sitting now with the source, that's you. So what do you think the public should understand right now about SARS and also monkeypox coming? What do we know about 2.5 years into this?
Well, I guess people talk a lot about post-pandemic, and I don't think anyone's told the virus that it's post-pandemic. So I feel like we're all really tired, and we all want our lives back and normalized again, on the one hand. And on the other hand, I don't think this virus is done evolving. And the way it is selected is for being either more infectious or being resistant to immune responses. Those are the two things that put pressure on the virus to change. So, we've still got to really keep an eye on it. And I think we have to be flexible enough to adapt. So that the vaccines are beautiful, they're extraordinary, and it was almost a miracle we were able to get them within a year. I mean, we've been trying my whole career to get an HIV vaccine, and we haven't yet. It's so variable, HIV, that it's a harder problem than the SARS-CoV-2. But, the virus can change and will slip through the cracks. And how that will manifest remains to be seen. So we had a big surprise when Omicron came through. It was very, very different than the viruses that had gone before it. And the vaccines are able to protect against severe disease, which is wonderful, but not able to protect so well against infection, which is why there was the huge, huge spike in infections. Right. And if that could happen once before, we don't really know what's next. So I think our exhaustion shouldn't let us be complacent. And a good way to think about this is if we need to respond, if the virus is changing in such a way that we need to respond, to be open to that. And, not just refuse to go back, but think, how can I protect my neighbors and my community and my family? What do I need to do? So it might be get a new vaccine if a new vaccine is warranted. It might be going back to wearing masks like this wonderful film crew is all doing. Thank you so much. But it's not that hard to wear a mask. And if you're in an upsurge and that upsurge is being tailed by hospitalizations and deaths, we need to be able to be agile enough as a society to go back for maybe just a brief period. And that kind of thing seems to be very difficult to implement, or that kind of agility seems to be hard for us. So I guess I wish that there would be more of a public health message going forward saying we need to respond to the virus and be aware, and maybe we'll really be lucky and we really are moving into post-pandemic. But if we're not, we have to be open and able to respond.
And I know it's still early, but do you have any hunches about what monkeypox is going to look like?
Well, I've been working on monkeypox for the last 2 weeks, actually, because
this is
it's
really
about
fresh,
2 weeks
fresh
old.
off the press. Okay, yes.
Right,
Yeah.
yes. And monkeypox is quite a different virus. And now people are saying that it evolves very slowly. But in fact, what's happened with monkeypox is it's a virus that is a distant cousin of smallpox, which, eradication of smallpox is one of humanity's greatest triumphs. That was a terrible disease. And the whole globe had to get together with a vaccine program to eliminate this horror from the earth. And we did that successfully. Monkeypox is not as severe as smallpox, but it's a quite unnerving disease. And historically, it's not really passed human to human. Right. So there have been cases of in Central Africa, there have been cases in West Africa, and they're usually thought to be from animal to human. And what's different in the last few weeks is it's clearly going human to human, and it's become really globally sampled, and the numbers are going up quite quickly. So it's not an airborne virus like SARS-CoV-2. It's you pass it by contact, and we don't even yet really fully understand how the pathology of this disease is going to manifest. But I noticed the first monkeypox virus sequences I looked at, there were just a couple out. You could see a pattern of mutation in these viruses, which was very like a pattern of mutation I was very familiar with from HIV. So one of the sequences was from the CDC, and I called them and talked to them about this, and they hadn't noticed it yet, but since that call, other people have noticed it and posted it. So it's now, I think, pretty well established. It's mutating by a mechanism that our innate immune response calls up. So it's a way we have of protecting ourselves against a new viral infection, and it causes mutation in genes. And that mutation has a specific pattern. So with monkeypox, since 2017, there have been some cases in Nigeria, and then there have been isolated cases in the UK and in Singapore, so 2018, 2019, and then now this surge of cases, and they all are almost entirely mutating by this human mechanism. So their evolutionary pattern is really different. And that means that tracing the epidemiology and time of origin, like you can usually do by looking at evolutionary patterns, you can't really do with this virus, at least not right now with this set. And we don't know why this particular brand of monkeypox that's expanding has this pattern of mutations. We just know it does. So it remains to be seen. Why is it? How has it changed? Is the virus that changed? Is it the host? Is it us responding to it that's changed? We have to figure that out. But it's definitely a really distinctive and interesting mutational pattern. So we're, I believe, submitting that paper today.
So your plans to retire keep getting put off. like your work does not seem done.
No, I don't think it's done. Yeah, I had planned to retire in June of 2020, and I was all set. And I passed my HIV work along to really wonderful younger investigators in my group that are magnificent. So I was feeling good about it, and I was going to help them out a little after I retired, 20 hours a week and write a book. And that was my goal. But that's not what manifested. What manifested was thinking, oh my God, I think I can be useful here. So when SARS-CoV-2 came out, I started frantically writing grants because I have to pay for my work and my staff to be able to work on SARS-CoV-2. So, February, March of 2020, writing grants, trying to prove it was evolving when the world was saying it wasn't, and I knew it was because of being able to see it in the data.
Right.
So so rather than retiring, it's been 8 to 12-hour days, 7 days a week since February 2020, actually.
And how long do you think you'll keep going at this clip before returning to your book?
As long as I feel like I'm useful.
Yeah. And the book, what is?
Well, so before SARS-CoV-2, actually, I can remember the date. It was November 9th, 2016. You might remember what happened then. President Trump had gotten elected, and I felt like I wasn't doing my job as a citizen. I felt like I was very intensely focused on my science, and I wasn't finding ways to share my science. And I think that a lot of us do this because science is hard. We have to work very hard and rigorously and focus. So asking people who are doing that to also take time to talk to community is difficult, and not everyone has that aptitude or enjoys that. But I actually do really like talking to people. And I felt like I wasn't expressing my work to the community. And I work in evolution and I work in vaccines. So these are two of the hot button topics, right? And I wanted to be able to explain to people how they worked. I wanted to be able to explain how beautiful the immune system is, what Ellen Rothenberg gave me years ago, to people who were citizens, not STEM students in high school, who were citizens who had to vote. And who had to teach their own families about vaccines. So I started doing that. I, gave March for Science talks, and I gave community talks and library talks and talks to teachers. So I was doing this while I was working. And I wanted to try to write a book that would express partly the beauty and partly the practical nature of what we can learn and what our science can do for us in a way that people could digest and understand if they wanted to take the time to read it. I had to stop that when I was doing SARS-CoV-2 because that took back the focus. I mean, I can't even Zoom call now, this kind of thing.
Right.
But I was doing that just prior to that, and it turned out to be really quite wonderful. People kept saying, but you're going to get anti-vaxxers in the audience, and in fact I did. And they were great. It was really good to talk to them.
So what was that conversation like?
My favorite was, it was actually at the March for Science. So I gave my talk and I read it because I didn't have slides. I couldn't used to, right? So I was reading my talk. And a woman came up to me afterwards and she gave me the most wonderful hug. And she said, I'm a grandmother and I have 12 grandchildren and none of them are vaccinated because my daughters and I all don't believe vaccines are healthy for them. We worry about autism. We worry about other aspects. And she said, I'm not sure that we should be doing this anymore. can you give me your talk? And I'm going to have a family meeting and we're going to discuss it at the dinner table tonight. So I handed her my talk.
Wow.
Yeah, so that was my very favorite, but that was, yeah, it was wow. It was just wonderful.
Grandmothers again.
Grandmothers, yeah. So anyway, I don't ever know what happened. She never got back in touch with me, but I had some really wonderful conversations with people who are in the anti-vax movement and active in it, and who are interested enough to try to hear the other side and were curious. And they were good people trying to do the best for their family, but they didn't have the right information. And so it was lovely. And I think the other way of was trying to be more active was the legislature in New Mexico is got this wonderful thing where citizens can speak. And that's, I think, even maybe unique in New Mexico, because you can stand up and have your 2 minutes on any issue. And we had for a while a Republican governor who wanted to take evolution out of the school curriculum, who wanted to take vaccine out of the health class curriculum, nutrition out of the health class curriculum. It was, and also the age of the earth out of any scientific conversation. So, you know, this was just so wonderfully inspiring. I started going to legislative sessions where I had something to offer, and that was one I wanted to speak at. And not only me, but nuns, librarians, school teachers, school children, business owners who wanted to attract good business to New Mexico. Everyone was at this meeting. There were hundreds of us all getting our 2 minutes about why we wanted good science taught to our children. And almost nobody on the opposition came. So, you know, you have these experiences and you realize how much people care, and it's really quite beautiful. So my book was going to be to try to have that, I guess what my father would have wanted, trying to express through writing and sharing, aiming for truth and sharing that with people. Yeah, so that was the book. Half written, maybe someday.
Wow, that's so fantastic, and thank you so much, Dr. Korber, for talking to us today, and can't wait to see your work mutate and evolve.
Thank you.
We're back. It's so fascinating. So, Dr. Korber, are you ready to take some questions from our audience?
Yes, I certainly am. Yeah.
And I just want to remind the audience that if you want to send questions, you can message one of the hosts. You can message me directly, but keep it nice. Yeah, you can message me directly, I believe, through the chat. And so we'll answer those as we go. But in the meantime, I'm going to go from some questions from short to long. Dan Fain, PhD in 01, which always seems so new to me, anything goes. What vaccine technologies are most promising for improved effectiveness at preventing long COVID?
Oh, well, I'm actually not technically sure how to answer that question, except that I've been hearing early reports that perhaps the new combination is more effective at long COVID. I think we'll have to just see what unfolds with that. It's going to take time to learn. But I think that the more potent immune responses, longer lasting immune responses are going to help. Just help us control the disease in our own system. And I don't think that we know exactly which components of the immune system are best for preventing long COVID. So it could be T cells are really important, for example. And the virus can persist in the body a bit. And you can, so that's, part of long COVID. So things that are good at eliminating that will be better. And I think it's going to take a few more years of study, actually, literally, to really sort that out. But it's a very good question. I'm sorry, I don't have a better answer for you.
Well, time will tell. It's after all long COVID, so long. This comes from John Horn, BS 1976 Chemistry and Chemical Engineering. His major was chemical engineering. Please, and this is, I guess, a bigger question. Please provide your thoughts on the origin of COVID-19. Is it man-made? Have you ever felt that the media, and we talked about this a little bit in the conversation, but I think it's asked in another way. Have you ever felt that the media or the government interfered in your ability to communicate on the science related to the virus? Would your funding ever be contingent on your public comments?
Well, so that's a couple of questions, but
Yes.
I think that there have been really beautiful and careful studies done now, suggesting that really the marketplace was the origin of this virus. There are extremely detailed epidemiological studies, and those are recently published, and I think they pretty much put it to rest. It's very, very solid case. Back in March of 2020, I started working with colleagues to try to look at the origins. And these viruses recombine naturally a lot. So if you look at related viruses in bats and pangolin and other animals, you can see the recombination events. So to me, from those very first moments, from the very first study, it was quite plausible that this was a natural event. I didn't see any reason to revoke necessarily that it was a laboratory event, even that early on, because it could have certainly been a creation of just the natural history and the movement through animals. So I think that when things were still uncertain, it was actually maybe even, you know, it was in the interview that Sandra and I had earlier, we talked a little bit about tweeting and sort of making pronouncements that were speculative. And I think that happened a lot in the early time before we really had clear data. And the scientific community was speculating, and the reporters were repeating that speculation. And passions got inflamed about the notion of whether it was laboratory or natural origin. And I think that probably was detrimental to cooperative science between China and us. Things became heated and political and difficult to work together, which was a shame because they had done so much research on bats and on related coronaviruses in China in the natural population. And we had cooperative efforts ongoing because of the first SARS epidemic many, many years ago. That was established. People are always afraid this was going to pop up again. There's another disease called MERS, which is another related virus that's moved into humans. We have other kinds of coronaviruses that are in us that cause common cold and stuff. So this has always been a concern that we might have a pandemic coming from a coronavirus. And China has done incredible research on this, and we worked with them. And it became very difficult to work together as a consequence of the heated and very negative discussion. So, you know, I have no problem with trying to pursue the origins of the virus, but as you get more data, and it's pointing towards one direction, it becomes sort of time to accept that notion, I think, and the data is really solid now. And even in the early days, to get so heated and angry about what was speculation was unfortunate, to say the least.
Yeah, a real distraction. Okay, another question from Hatisuslu, BS 96, Economics, is a Strategic Initiatives Manager at AbbVie, joining from Istanbul, Turkey. And it's so fun on these Zoominars, if you will, because it's so international, the Caltech audience. So, and this, again, I'm going to roll through these questions. It is a multi-question packet here. So I'll read it aloud and you can how successful are the corona vaccines? Any hope for HIV vaccine? Well, I'll roll out all the questions and then you can go for it's many. What is it about
Okay.
HIV and coronaviruses and their mutations that make vaccines design a challenge? Are there any other viruses that are currently out of the spotlight that we need to plan for? I'll let you digest those first.
Okay,
So
well, I guess I'll go with HIV first. HIV is incredibly variable. And part of the difference between HIV and our coronavirus infections with SARS-CoV-2 is that we clear influenza virus. HIV is chronic and it's a lifetime infection, so you can treat it and you can have a great life with good therapy if you're fortunate. But all the time that it's been in individuals, it can be evolving under immune pressure, and you kind of have this cat and mouse game going on, where if you get infected, you make an immune response, the virus evades that immune response, but the infection persists, you get a new immune response, and then the virus evolves. So it's this incredible evolution within every individual. And that evolution is driven by immune escape to a large extent. So now we've got to make a vaccine that's able to contend with this incredible diversity. And a lot of the diversity has been generated by immune escape. So we've got this really terrifically difficult challenge with HIV for making a vaccine that can actually prevent infection and manage to block all of the myriad viruses that you might encounter in the world, because it's just so incredibly diverse. And I think we're the efforts right now are really focused on some very elegant, very complicated strategies that may be able to overcome this by training antibodies to see breadth, to see a lot of different variants. And we are taking steps in that direction. Whether it will succeed or not, I'm not sure. But I think there's a chance that it might, and the immunology that we've learned along the way and the vaccine deliveries that we've learned along the way are helpful science anyway. So hopefully we've got a possibility of an HIV vaccine in the future. I'm not sure if it will be realized, but the research doesn't go to waste anyway. And an example of this is that the HIV research that people were doing using mRNA vaccines that went into Moderna and Pfizer to try to get better deliveries for HIV. This had us really had us really primed to be able to make a SARS-CoV-2 vaccine when it came up quickly, because there had been just a massive amount of research on the deliveries, on the immune responses, on how to do neutralizing antibody tests. So we knew that kind of thing going in, and we were ready to get this vaccine in the year because of the other research. And something like SARS-CoV-2 does evolve a lot more slowly, and it clears, and so you don't have this continuous cycle in every single infected individual, which is why the ancestral Wuhan virus that we have in our vaccines up until this point has been so effective, because it's at least it's been able to make good immune responses that are certainly able to help have a better course. And until Omicron, we're pretty also could help against infection. And I think that we're getting a world now that's also been infected so much. We're getting good immune memory in our world. And when you couple the immune memory of a real infection with the vaccine, the immune response is even more potent and more powerful. So we've got a potential of a population that's getting much more resistant to this virus, which is a great thing. But there's I guess there's a lot to be learned, and one of the things that many groups are working on right now, which I'm also helping on this kind of a concept, is to try to get antibodies that would work against a whole spectrum of coronaviruses. And so those might be beneficial in two ways. One is it would be harder for SARS-CoV-2 to evolve away from them, because you'd have captured these really conserved bits that are present in many coronaviruses. And so it's more difficult to sneak away from something that has to be conserved across viruses. And two, if there's another coronavirus introduction, we'll be prepared with an antibody that could react well with it, whether it's vaccine induced or whether it's antibodies that you create in the freezer to use as a therapeutic. So these kinds of strategies to find what are the conserved bits and what are the bits of the virus that it needs to maintain its biology, to maintain its phenotype. Can we target those with immune response, and one, be prepared for new pandemics, and two, maybe have a good set of reagents to use against the current pandemic. That's an interesting avenue people are taking, and that I'm excited to be part of.
So Charles Spence, PhD 02, Engineering Applied Science, coming from Arcadia, California, a little closer to where we are now. This is also kind of a longish question, but the key part of it is, can we detect SARS-CoV-2 or similar viruses with the nasal swab from a person before they become infectious, aka before they are producing so many viral particles that it increases the rate of these particles being expelled when they breathe, hence infecting others? There are other parts of that question, but that's
right. So there's certainly more detection technologies that are being advanced and tested and promoted, and so I think people are trying to get more and more sensitive
Yeah, and
approaches.
if I can
Yeah.
relate a little bit more of the lead up, that I understood that affinity spin columns were the best way to purify DNA and RNA samples. With a nasal swab that is placed in a detergent solution, you know the number of viral particles you need to enable capture of at least one copy of the RNA by a spin column that can be eluted off for PCR.
Well, you're out of my expertise, so I'm not gonna be able to give you, but I do know colleagues working on those kinds of questions and striving to get better technologies, cheap and better technologies.
Yes, that is the joy of our Caltech audience. Often, yes, we see, yes, RT-PCR systems,
Right.
viral particles, et cetera, et cetera. You know, and I think it goes back to the microbiome is a hot topic. Now we see a composition of microorganisms carrying millions of unnamed viruses, particularly in our guts, and they're responsible for many chronic diseases. Yes, your thoughts. It's how you see it.
Well, it's an incredibly valuable new area, not new, but getting richer continuously. And I think this balance, our bodies are these amazingly intricate ecosystems supporting bacterial and other kinds of life forms within us. So they win out in terms of sheer numbers. There's more of them than us within this little skin suit that I'm wearing. So I think understanding this is really incredibly important, but it's also they're part of our health. They're part of this is not only pathogens, but symbiotic relationships, and we need to understand those better. And sometimes we can have ill health situations arise from trying to clear antibiotics that try to clear too much when we actually need to be these complicated systems.
Yes, and I think that's a great way to end. And I love your use of the phrase "skin suit." And Harrison Leong notes some viruses are good, and so yes, we're a stew of all of these pieces under our skin suits, and it's so fascinating to hear you talk. And congratulations on your Distinguished Alumni Award.
Thank you.
Thank you, Dr. Bette Korber, very much for joining us. Thank you. And yes. Oh, so that concludes our interview. I've been Sandra Tsing Loh and still am for the Caltech Alumni Association. Just a reminder to you all that Alumni Weekend Reunion kicks off with a Behind the Book event on Thursday, October 20th at Beckman, with me actually talking about Caltech and fathers. So come on down with Samantha Dunn of the Southern California News Group. Register for the entire weekend events on the Caltech Alumni Association website. Join us for Dr. Sudhir Jain on Saturday, November 5th at 6 p.m. He will join us from India. Registration is open. Do you know a techer to recommend for our 2023 Distinguished Alumni Award? Nominations are open. And while you're there, check out our Techer Alumni Tours. See the world with other techers to Patagonia, Paris, Egypt, and South Africa. What are you waiting for? Join us. Okay, so on behalf of the Caltech Alumni Association and Caltech, I'm Sandra Tsing Loh saying thanks for joining us today for lunch. And remember, Caltech motto, the truth shall make you, let me say that again, the truth shall make you free. Yeah, because I thought I had been told that